Okay, here we go. It's showtime. Okay, as people are trickling in little by little, we're going to do a little bit of recall from yesterday's topic. Yesterday, we worked with some historical studies that led us to the point where we understood that DNA was the genetic material. So your biozone today deals with Griffith and Avery McLeod-McCarty. And so the two historical studies both dealt with mice and Streptococcus pneumonia. So you want to take a minute to read the background knowledge, remind yourself about those studies. And the questions that follow are onto the next page, and there are only 3 questions. So the big idea here, lots of studies went into our ultimate understanding of DNA as the genetic code. We looked at Griffith's experiment with the rough and smooth strains of Streptococcus pneumonia. The smooth strain was deadly, killed off the mice. The R strain was not. When he combined the two, the R strain became deadly, even though he had heat killed the S strain. Then you had Avery McLeod-McCarty trying to figure out. Avery McLeod-McCarty were trying to figure out what the transforming factor was for Griffith. And they one by one used enzymes that destroyed different components of the bacteria. And they found when they destroyed the DNA, they did not get transformation. So Griffith was not expecting transformation to happen. He was expecting that when he combined the R and the S strain, after he had heat killed the S strain, he thought that would take away its deadly component. So he was kind of surprised that there was transformation. So he was expecting the R plus S to create a living strain that did not kill the mice. Avery's experiment concluded that DNA was the transforming factor. And how did it build on, so that DNA was the genetic information, and how did it build on Griffith's experiment? Because Griffith's experiment came to an end with transformation, and then Avery figured out what was causing the transformation, and that was DNA. Yesterday's studies led us to the point where we understood that DNA was indeed the genetic material. So Hershey and Chase, the final study, kind of solidified that idea, right? So now that we know that DNA is the genetic information, we know a little bit about its characteristics. We know that it's long and thin, and it has 4 nitrogen bases: adenine, thymine, cytosine, and guanine. We know it has a phosphate group and a 5-carbon sugar. So we know a bit about its chemical makeup. We want to know now, what is its structure? Okay, so the next set of studies goes into determining the structure of DNA. So we start with Maurice Wilkins and Rosalind Franklin, and they worked with something called X-ray diffraction. So you've probably had X-rays taken of your teeth. So you get a little bit of the idea that you're shining these X-rays onto a substance, right? Well, in this case, the substance is this thin, stringy, kind of snot-looking DNA, right? So this is a sample of DNA. Next week, we'll do an extraction lab in class. And let's say this is the DNA, and they're going to shine a light. Ooh, if I was quick, I would get my light shining from my phone. So they're going to shine a light or something on the substance, right? And then the X-rays will pass through, and at certain points it can't, right? So where it's hitting the DNA, it won't be able to go through. So it passes through and it lands on a photographic paper. So that's what you see here, that photographic paper. So where the X-rays are able to pass through, whereas there's transmittance, it leaves marks. Okay, so this is like actually the gaps in DNA. So the X-ray diffraction led Watson and Crick, who ultimately determined the structure of DNA, to determine that DNA was a helical pattern. This X in this picture, this is photo 51, a very famous picture. The X in this pattern told them that the DNA was helical, like it wrapped around itself like a twisted ladder. They also could see by the number of specks that there was 2 strands wrapping around each other, because we didn't know if it was 1 or 2 or 3. And they could determine distances based on the amount of space in between markings. So Rosalind Franklin and Maurice Wilkins worked with the X-ray diffraction. The pictures were actually taken by Rosalind Franklin. So the picture that really determined the shape and structure of DNA was under the hand of Rosalind Franklin, who died at age 37 because of cancer due to all of her work with X-rays. The rest of the cats, they all get a Nobel Prize because of their, ground-shattering discovery, which they couldn't have done without without Rosalind's picture. But they don't award Nobel Prize posthumously after you've died. So Rosalind was not one of the members of the Nobel Prize. So another person that that played a really big role in Watson and Crick figuring things out was Erwin Chargaff. He did chemical analysis of the different molecules in DNA. And he found that adenine, A, and thymine appeared in equal amounts. And they were in equal amounts in a certain species. So like maybe adenine and thymine make up 38% of the human DNA structure. That's going to be pretty consistent with all humans. And maybe in a dog, it's 34%. So it's consistent within a species and it's equal to each other. Guanine and cytosine always occur in equal amounts as well. So for every A, there is a T, and for every G, there is a C. So they appear in equal amounts. Watson and Crick took that and said, if they appear in equal amounts and there's 2 strands, they must be bound to each other, which in fact they are. So we should be well versed in the structure of the nucleotide. DNA is made up of, remember, it's a polymer of nucleotides. We learned this in chapter 6. So this is not the first time we've seen this structure. So we've seen a phosphate, a sugar, and a nitrogen base. So the sugar is deoxyribose, thus the name deoxyribonucleic acid. So we want to know that, and we want to get a little bit more familiar with these nitrogen bases. So the nucleotide is made up of those 3 things, the phosphate, the sugar, and the nitrogen base. You are going to be creating a model of DNA next week. So you can start thinking about what could you use for each of these components of the DNA model. What could you use for a phosphate? What could you use for the sugar? What could you use for your nitrogen base? There's 4 different nitrogen bases, so we have to have 4 different things to represent the bases. Okay, Watson and Crick took the picture, photo 51, decided it was a 3D double helix, 2 spiraling strands wrapping around each other. And when they made their model of metal and cardboard, the first time they made it, they were way off. Like the structure wouldn't have held up. So scientists kind of laughed at them and they were told to quit playing with their models and they weren't allowed to work on DNA anymore because they were a bit of an embarrassment to the Cavendish. But they got back at it and they made some corrections and then they invited the scientists back and the scientists were just, mouth dropped in awe. And that it was so beautiful that it had to be, like it so perfectly fit all of the experimental data that it was almost immediately accepted. And they looked at it and they said, so that's how it is. So these characters, Watson, Crick, and Maurice Wilkins, were all awarded the Nobel Prize in Medicine due to their discovery of the DNA structure. Rosalind Franklin, had she been alive, I'm sure she would have been in that list. So looking at our structure, we can see the 2 strands of DNA. The other thing that the model explained was how DNA could replicate itself and get exact copies time after time. So the fact that the 2 sides are complementary, meaning one side determines the other side, that explains how if you opened up the 2 strands and you made a copy of one, you're going to get the other. Right? So one side determines the other side, complementary. C has to bond with G. So if you took that G away, you could only replace it with another G. Notice some of these are double rings and some of these are single rings. Notice some of these have double bonds, some of them have triple bonds. This was another part of determining the structure. They needed a structure that was the same width the entire way, right? If they had 2 of the double rings, you would have had wide spaces. If they had 2 of the single rings, you would have had skinny spaces. So the only way to get the DNA consistent was to pair a big with a little, or a double ring with a single ring. So this explains how the 2 sides come together. These bonds in the middle here, these are hydrogen bonds. Hydrogen bonds are very weak bonds, which again aids in the understanding of how replication can occur. If it's a weak bond, we can break that bond, separate the 2 sides, and gain access to the code where we can make a copy of the other side. Okay. So now we know all of the chemical components. We want to be able to put together our model of DNA. We need to know what DNA stands for, deoxyribonucleic acid. So the D, N, and A. We're going to have something represent deoxyribose. We know that DNA contains the genetic information. Hershey and Chase solidified that piece of information. We know that it's composed of 2 strands of repeated units called nucleotides. 2 strands, helical. Rosalind Franklin gave us that. Looks like a twisted ladder. Those nitrogen bases that Chargaff works with. The adenine, the thymine, the cytosine, and the guanine belong to 2 categories of molecules called purines and pyrimidines. So in this picture here, we can see the purines are these double ringed structures. So in your model, you're going to want something to be a double ring, or you're just going to want something that is bigger, right? And then your single ring are your pyrimidines. So again, a single or something smaller. Like you could use a big marshmallow and a little marshmallow. That would work. If you work with marshmallows, let them set out the day before because they're too soft to work with otherwise. So purines are adenine and guanine. So a purine will bond with a pyrimidine. Ag stands for silver. So if you wanted to remember pure silver, that could be a mnemonic for you. The pyrimidines are the single rings, and we'll notice that cytosine and thymine both have Y's in it, and so does pyrimidine. So that could be a mnemonic for that. Okay. So you need to know which 2 belong to the purines. That will be a test question. You need to know which 2 belong to the pyrimidines. That will be a test question. You need to know which purine bonds with which pyrimidine. That is a test question. So the hydrogen bonds hold them together. We have 2 bonds between adenine and thymine. We have 3 bonds between guanine and cytosine. So in your model, you'll want something to represent your bonds. Maybe you use toothpicks, and you use 2 toothpicks between thymine and 3 toothpicks between cytosine. Notice there's 3 bonds between G and C and 2 bonds between A and T. Even if these weren't labeled, I would be able to tell what is what, because I know the double rings are to adenine and guanine. So that limits my thought there. And then because it has triple bond, I know it's got to be guanine or cytosine. So guanine fits into both those categories. So that is how I would figure that out. So to say that the 2 are complementary says one side determines the other. So if you have GTC, the only possible match is CAG. So take a moment, write me a note in the chat. What would the other strand be? If this strand is ATC, CGAAT, what would the other strand be? So A bonds to T, T bonds to A, C bonds to G, G bonds to C, A bonds to T, T, and T bonds to A. Very good. Any questions on today's edition of the lecture? So we're going to do this Wiser Me before I turn you loose to finish yesterday's recap. So yesterday we did 1 through 8. Today we're going to finish up with 9 through 12, 13, 9 through 13. So before we do that, you want to do this. You have word choices down here. Be sure to use the same capitalization and spacing. Matching. Use your base pairing rules to drag the matches. You're going to write capital letters in here to represent the corresponding letter and label the S and P. This is referring to all three pieces together. Here's a little bit of math. It's going to add up to 100%. So it's a little puzzle. Remember, adenine and thymine are in equal amounts. So if adenine is 15, so is thymine. That makes 30. That means there's 70 left. Cytosine and guanine together will equal 70. And they are in equal amounts. That'd be 35. You got one more to do that's a little trickier.